Have you ever stopped to think that, well, the light all around us might actually hold hidden stories, secrets about the world we can't normally see?
It's a fascinating thought, isn't it?
Yeah. And imagine a tool that lets us, actually peer into that invisible world
Yeah,
and quantify what's there.
precisely.
That's exactly where we're going today.
Yeah.
We are taking a deep dive into UV visible spectroscopy. Now, it might sound a bit complex,
Uh-huh.
but it's this incredibly powerful technique. It uncovers surprising things about, well, everything from DNA purity to the quality of medicines.
Absolutely critical stuff.
So our mission, really, in this deep dive is to give you a shortcut, a way to understand how light interacts with matter without getting totally lost in jargon.
But still get those aha moments.
Exactly, those moments.
Sure.
And we've pulled our info from actual lab demos, expert guides,
Mm-hmm.
so you get the theory and the practical side.
Yeah, and at its core, UVV spectroscopy, it's really a technique that measures how light interacts with a sample. Specifically light in the ultraviolet and visible plans of the spectrum, and the instrument, the machine we use, is called a spectrophotometer.
Spectrophotometer. Yeah.
And the basic idea is actually quite elegant. It's all about measuring how much light the sample absorbs.
Or transmits.
Or transmits, exactly. And that number, that measurement, it unlocks really critical information like concentration or even what the substance is.
So, okay, light interacting with matter. How does that actually work? Like, fundamentally, how does this invisible thing become something we can actually measure?
Right. That's where the real magic of spectroscopy kicks in. So when light passes through your sample,
Mm-hmm.
the electrons inside the molecules, they absorb specific amounts of that light energy.
Specific amounts of
Yeah, and this energy causes something we call an electronic transition. Basically, it kicks an electron from its comfortable low-energy state, its ground state,
Right.
up to a higher energy excited state.
Right.
And the crucial thing is the amount of energy needed for that jump, that transition, it's unique for each molecule.
So different molecules need different energy jumps.
Precisely, which means they absorb different specific wavelengths of light. And this leads us to a really key concept, lambda max, or Omax.
Lambda max.
What's fascinating is that every compound has its own peak absorption wavelength. It's Omax, the specific wavelength where it absorbs the most light.
So it's like a fingerprint.
Exactly, a unique spectral fingerprint. Finding that Omax is, well, it's absolutely vital for identifying what you've got, for characterizing the compound.
Okay, understanding how it absorbs is one thing, but how do we turn that into a number, a measurement? How do we use that to find out, say, the concentration?
That's precisely where the Beer-Lambert law comes in. It's kind of the cornerstone, the mathematical rule book for UVV.
Beer-Lambert, named after.
August Beer and Johann Henri Lambert. They figured out this relationship.
Okay, so what does it say?
It states that the absorbance of a solution, how much light it blocks, is directly proportional to two things.
Two things.
One, the concentration of the substance that's doing the absorbing.
Makes sense. More stuff, more absorption.
Exactly. And two, the path length. That's just the distance the light has to travel through the sample.
Okay, the width of the container, basically.
Pretty much. The formula is actually quite simple. Absorbance A equals...
A equals epsilon CL. Okay, break that down.
Right. So epsilon is the molar absorptivity. It's a constant that's unique for each substance at a specific wavelength, like part of its fingerprint.
Got it.
C is the concentration. That's often what we want to find out. And L is the path length, usually 1 centimeter because most sample holders or cuvettes are 1 centimeter wide.
So higher concentration means higher absorbance. It's directly linked.
Directly linked. But, and this is a really important practical point, the absorbance readings, sometimes called OD for optical density, they need to be in a certain range for the law to hold true. Ideally between, say, 0.2 and 0.8.
Why is that?
Well, if the absorbance gets too high, usually above 1, things can get complicated. You run into nonlinearity, meaning your graph of absorbance versus concentration won't be a nice straight line anymore. It'll start to curve.
And that messes up the calculations.
Exactly. You can't accurately determine concentration from a curved line using this simple law. And that's why very often in the lab, you'll dilute your sample.
To bring the absorbance back into that sweet spot.
Yeah.
That linear range.
Precisely. Get it between 0.2 and 0.8, and you're usually good to go.
Okay, we've got the principles, the math, but how does the actual machine, the spectrophotometer, make all this happen? What's inside the box?
Right, the spectrophotometer. It's the workhorse that brings it all together. Let's break down the key parts.
Okay.
First, you obviously need light. So you have light sources, typically two.
Two. Why two?
Because you need different lamps for different light regions. There's a deuterium lamp for the UV range,
Okay.
and a tungsten filament lamp, kind of like an old school light bulb, for the visible range.
Got it. UV and visible covered. What's next?
Then the light, which is initially all mixed wavelengths, hits the monochromator. You can think of it as a wavelength selector.
Ah, like a prism or a grating.
Exactly. Its job is to pick out just one specific wavelength of light from that mix. It turns polychromatic light into monochromatic light.
One color, basically.
Mm-hmm.
Or one wavelength.
Precisely. And that single wavelength is what passes through your sample. Then, finally, there's the detector.
Which does?
It measures the intensity of the light that actually makes it through the sample, the transmitted light.
Okay.
It converts that light signal into an electrical signal, which then goes to a display or computer, and that gives you your absorbance reading.
Simple enough in concept. Now, what about holding the sample? You mentioned cuvettes.
Ah, yes, the cuvettes. These little transparent containers are surprisingly important, and you have to handle them right.
How so?
Well, first, the material matters a lot. If you're doing analysis in the UV range.
Below 400 nanometers.
Right. You must use quartz cuvettes.
Must use quartz.
Because regular glass cuvettes, or even plastic ones, they actually absorb UV light themselves.
Oh, so they'd interfere with the reading.
Massively. You'd get false high absorbance. So quartz only for UV. For visible light, though, glass or plastic is usually okay.
Good to know. And handling them.
Very carefully. These things can be expensive, like really expensive. A good pair of quartz cuvettes. Hundreds, maybe thousands of dollars.
Wow. Okay. Handle with care.
Definitely. And always hold them by the frosted sides, the opaque parts. Never, ever touch the clear, transparent sides where the light beam goes through.
Fingerprints.
Exactly. Fingerprints, smudges, dust. Anything on that optical surface will block some light or scatter it, giving you inaccurate results. Always wipe them clean, gently, with a lint-free wipe like Kim wipes before putting them in.
Makes sense. What about filling them? How much sample?
Good question. You need to fill it enough so the light beam passes cleanly through the liquid. Minimum half full, but ideally maybe 2/3 full. But you definitely shouldn't fill it past the little line or mark they sometimes have. Keep it below the optical path line.
Bubbles. I imagine bubbles are bad.
Bubbles are definitely bad. That raises a really important point. You need to make sure your sample in the cuvette is bubble-free.
Why?
Because if a bubble happens to be in the light path when the measurement is taken, it'll scatter the light everywhere. It can seriously mess up your absorbance reading. So, check for bubbles. Tap gently to dislodge them if needed.
Okay, cuvettes prepped, sample loaded, no bubbles. How do we actually run the measurement? You mentioned different types of machines earlier.
That's right. There are two main types of spectrophotometers you'll encounter: single beam and double beam.
Okay, what's the difference?
A single beam instrument has, well, just one place to put a cuvette.
Simple enough.
Yeah. So you first put in your blank solution, we'll talk about that in a sec, and
Right.
zero the machine. Then you take the blank out, put your sample in, and measure it.
Sequentially. Blank, then sample.
Exactly. The potential downside is, taking things in and out, maybe the calibration isn't perfect between the blank and sample reading. Small variations can creep in.
Okay, so what's the double beam?
The double beam spectrophotometer has two cuvette holders, one for the sample and one for a reference, which is usually your blank.
Two at once.
Right. Inside, there's a beam splitter that divides the light beam. Part goes through the sample, part goes through the reference simultaneously.
And the advantage there is?
Better stability and accuracy. It's constantly comparing the sample to the blank in real time. It corrects for any fluctuations in the lamp intensity or detector sensitivity. It ensures proper calibration throughout the measurement.
So double beam is generally preferred for high accuracy work.
Generally, yes. It allows a direct, simultaneous comparison, which is usually better.
You mentioned the blank solution. What exactly is that and why is it so important?
Okay, blanking. This is a crucial step. The blank solution is basically everything in your sample except the specific thing you want to measure, the analyte.
So usually the solvent?
Often, yes. If you dissolved your sample in water, your blank is water. If you used a buffer, the blank is the buffer. It contains all the matrix components.
And its purpose.
Two main things. First, it calibrates the machine. It tells the spectrophotometer, okay, this is my baseline, this signal from the solvent. Ignore it. Set this to zero absorbance.
So you only measure the absorption from your actual substance of interest.
Exactly. It subtracts the background. And second, especially important in a double beam machine,
Yes.
when you do that initial zero calibration, you must put the blank solution in both the sample holder and the reference holder.
Both. Why?
because the machine needs to compare to set its 0 point accurately. If you only put a blank in the reference holder and leave the sample holder empty.
It thinks there's zero transmission there.
Right. It won't calibrate correctly. It won't get a proper reading. So blank in both for the initial zeroing on a double beam.
Got it. Blanking is key. Are there different ways to use the spectrophotometer once it's blank? Different modes?
Yeah, typically 2 main operational modes. Depends on what you need to find out.
Okay.
If you already know the wavelength where your substance absorbs maximally, it's Limax.
Like, salicylic acid absorbs best at 294 nanometers.
Exactly. Then you'd use fixed point measurement. You just tell the machine, measure the absorbance at 294 nanometers, and bam, you get your reading. Quick and easy for known samples.
Makes sense. What if you don't know the OMX, or you want to confirm it?
Then you use wavelength scan measurement, or scanning mode.
Scanning.
Yeah. You tell the instrument to measure the absorbance across a whole range of wavelengths, maybe from, say, 200 millimeter way up to 800 millimeter, or even 1100 millimeter.
And it plots it out.
It plots absorbance versus wavelength, and that graph, that spectrum, will show you the peak, the UMAX. It's essential for characterizing unknown samples or confirming the identity of something.
That's a fantastic overview. Theory, machine, modes. Now let's make it really concrete. One of our sources talked about an actual experiment analyzing salicylic acid. How would all this come together in the lab?
Okay, yeah, the salicylic acid experiment. That's a classic UV-Vis application. The main goal there is usually to figure out the concentration of some unknown salicylic acid solution.
Right.
So the chemicals involved are your salicylic acid, obviously. That's the analyte, the thing you're measuring. Then you need a solvent. In that case, they used 2-propanol, which is an alcohol, to dissolve it initially.
Why 2-propanol, not just water?
Good question. Salicylic acid is actually only sparingly soluble in water, doesn't dissolve very well, but it dissolves nicely in 2-propanol, which is a polar organic solvent.
Okay.
But then, interestingly, they use distilled water for making the final volume and for dilutions.
Why add water later?
That's to avoid an unwanted side reaction.
Yeah.
If you just used pure 2-propanol, especially over time or if heated, it can react with the salicylic acid, an esterification reaction.
Forming something else.
Exactly. It forms isopropyl salicylate. And that new compound has a totally different absorption spectrum, a different OMAX than salicylic acid. So adding water prevents that reaction and keeps your salicylic acid as salicylic acid.
Clever chemistry detail. And it's Lumex. You said 294 nanometers. Why there?
Right, 294 nanometer. That specific absorption is due to the electrons in its structure, particularly associated with the double bonds in the benzene ring and the carbonyl group. It's what we call a pi to pi star electronic transition.
Okay, fancy term. But basically, its structure makes it absorb light really well at 294 nanometer.
That's the gist of it, yes. That 294 nanometer peak is its characteristic fingerprint.
So how do you use that fingerprint to find the concentration of an unknown?
That's where the standard curve comes in. It's fundamental for quantitative analysis.
Yeah, standard curve, right.
You start with a stock solution of salicylic acid where you know the concentration precisely. Then you make a series of dilutions from that stock solutions with lower known concentrations.
Using that C1V1 equals C2V2 formula.
Exactly. You calculate the dilutions carefully. Then you measure the absorbance of each of these known concentrations at 294 nanometer.
Okay.
And you plot the results. Absorbance on the y-axis, concentration on the x-axis. If you've done everything right and you're in that linear range we talked about,
You get a straight line.
you should get a beautiful straight line passing through the origin. The equation of that line is basically y equals mx, where y is absorbance and x is concentration.
So once you have that line,
You just measure the absorbance of your unknown salicylic acid sample, find that absorbance value on the y-axis of your graph, go across to the line, and then drop down to the x-axis.
and that tells you its concentration.
Precisely. Or you use the equation of the line. Plug in the unknown's absorbance, y, and solve for concentration x.
Okay, that connects the dots nicely. Can you quickly walk through the practical steps like cuvette washing to getting the number?
Sure. So first step, always. Clean your cuvettes meticulously. Rinse them maybe twice with distilled water. If you suspect oily stuff, maybe rinse with ethanol first, then water.
Okay, clean is key.
Then dry them gently, outside only, with a lint-free wipe. Don't want scratches. Remember that story about the 50,000 rupee cuvettes? Handle them like jewels.
Right, no scratches.
Next, if it's double beam, load your blank solution into both cuvettes. Put them in the machine,
And run
the automatic zero calibration, or auto 000 EDO. Let the machine set its baseline.
okay, zeroed.
Now, Carefully take out only the cuvette from the sample compartment. Don't disturb the reference blank.
Right.
Rinse that sample cuvette once or twice with a small amount of your first standard solution or your unknown solution, just to condition it.
Get rid of any leftover blank.
Exactly. Then fill it properly, 2/3 full, no bubbles, below the line. Wipe the outside clean. Put it back in the sample holder. Close the lid.
And measure.
And measure. Record the absorbance reading. Repeat that process for all your standards and your unknown sample.
Seems straightforward, but needs care.
Absolutely. Precision matters at every step.
Now, labs aren't perfect. Things go wrong. What are some common problems you see with UVVs, and how do you fix them?
Oh yeah, troubleshooting is part of the game. A really common one is getting absorbance readings that jump around, fluctuating OD.
What causes that?
Usually it means there's something floating in the sample. Tiny particles, maybe some precipitate, or even microscopic air bubbles that haven't cleared. They drift in and out of the light path.
So how do you fix floaties?
The best way is usually to centrifuge your sample. Spin it down hard. The particles will form a pellet at the bottom. Then you carefully pipette off the clear liquid, the supernatant, and measure that.
Get rid of the moving bits. Makes sense. What about getting a negative absorbance reading? Is that possible?
It is. Now, if you get a slightly negative reading when you measure your blank during the zeroing step, that's usually okay. The machine is just setting its 0 point.
But for a sample?
If you get a negative absorbance reading for your actual sample, that's almost always an error.
What kind of error?
It usually means something went wrong with your blanking procedure. Maybe your blank was dirtier than your sample, or you measured the blank incorrectly, or maybe the sample concentration is truly 0, but usually it points to a procedural issue. You need to reblank and remeasure.
Okay, good tips. So we focused on salicylic acid, but clearly this isn't just for one chemical. Where else is UVVs a big deal? What are the broader applications?
Oh, the applications are incredibly diverse. It's a real workhorse technique. A huge area is purity assessment.
Purity, how?
Well, think about DNA or RNA in molecular biology labs. They have a strong absorbance peak around 260 nanometers.
Okay.
Proteins, on the other hand, especially those with aromatic amino acids like tryptophan, absorb more around 280 nanometers.
Right.
So, by measuring the absorbance at both 260 and 280, you can calculate the A260-A280 ratio.
And that tells you?
It tells you how pure your nucleic acid sample is. A ratio of around 1.8 for DNA or about 2.0 for RNA indicates pretty pure stuff. If the ratio is lower, it's just protein contamination. It's a super quick quality check.
Wow, that's neat. What about in medicine?
Absolutely vital in pharmaceutical analysis. Used constantly to test the purity of a drug substance.
Wow.
Every drug molecule has its own characteristic UVV spectrum, its own LOMX. If you run a sample and see extra peaks, or if the main peak is shifted, it's a strong indicator that there are impurities present, or maybe the drug has started to degrade. Quality control relies heavily on this. It's also essential for just monitoring things that grow, like bacterial cultures or cell cultures.
How does that work? You're not measuring a specific chemical there.
No, you're measuring turbidity or cloudiness.
Yeah.
As bacteria grow in a liquid culture, the broth gets cloudier.
Right.
You can measure this cloudiness called optical density or OD, usually at 600 nanometers where the cells scatter light but don't absorb much specifically. The higher the OD, the more cells are present. It's a quick way to track growth.
Clever. Any other big ones?
Definitely. Reaction kinetics, watching how reactions happen over time. If a reactant absorbs light at a certain wavelength and the product doesn't, or vice versa, you can monitor the change in absorbance over time to see how fast the reactant is being used up or how fast the product is forming. Great for understanding reaction rates.
So time-dependent analysis.
Exactly. And also protein estimation. There are assays like the Bradford assay where a dye binds to proteins.
And changes color.
And changes color, which means its absorption spectrum changes. The dye itself might absorb at, say, 465 millimeters, but when bound to protein, the complex absorbs strongly around 595 millimeters. So you measure absorbance at 595, and it tells you how much protein you have.
So many uses, from DNA purity to watching bacteria grow to checking drugs.
Yeah, and even things like analyzing components in beverages, checking for impurities in industrial chemicals. It's really versatile.
So after diving this deep, let's boil it down. Why does UV visible spectroscopy really matter? Why is it such a go-to technique? What are the killer advantages?
I think what makes it so enduring and valuable comes down to a few key things. First, it's generally non-destructive.
Meaning you don't destroy the sample.
Exactly. You shine light through it, you measure it, And usually you can get your sample back intact for other tests. That's huge, especially if your sample is precious or limited. It's non-invasive.
That's a big plus. What else?
It's a really quick technique. You can get a reading in seconds or minutes. Allows for high-throughput screening where other methods might be too slow.
Fast results, good.
It also allows for easy integration. It fits nicely into lots of different experimental workflows. And related to that, it's very user-friendly.
Easy to learn, easy to use.
Relatively speaking, yes. Once it's set up and calibrated, running samples is often quite straightforward, and the basic data analysis isn't overly complex.
Okay. Non-destructive, fast, easy to use. Anything else?
Yes. And this is a big one. It's generally cost-effective.
Compared to other fancy lab equipment.
Compared to many other analytical instruments like mass specs or NMRs, spectrophotometers are relatively inexpensive to buy and operate. The running costs are usually quite low.
So accessible power, basically.
That's a good way to put it. Reliable, versatile, affordable power to see the unseen. Imagine, instantly checking if a drug batch is pure right on the factory floor, or a researcher quickly confirming their DNA is good before a complex experiment. That's the kind of practical power UVVs delivers every day.
Okay, so you've definitely taken us on a deep dive into UV visible spectroscopy. We've gone from, just light interacting with
Molecules.
the electronic transition.
Right, to the Beer-Lambert law, letting us quantify things, to the nuts and bolts of the spectrophotometer.
Cuvettes and blanking.
Don't forget the cuvettes. And we've seen how it plays out in real examples like salicylic acid and its huge range of applications. It really is a critical tool, isn't it? Uncovering these hidden details all around us.
Absolutely, from research labs to industrial quality control.
So it leaves you wondering, doesn't it? What other hidden truths are just sitting there waiting to be unveiled, maybe just by looking at light a little differently?
It's a great question to ponder.
Well, thank you for joining us on this deep dive today.